Hello friends, welcome to Udan Career Institute once again. Today we'll continue with chapter 1 of class 12 biology, that is reproduction in organisms. In today's class we will focus on post-fertilization events. In earlier classes we have completed the first two events which occurs in sexual reproduction, that is pre-fertilization events and fertilization. In today's class, we will go through the third type of event that occurs in sexual reproduction, that is post-fertilization events. Now, what do you mean by post-fertilization events? Now, these are the events which happens in sexual reproduction after the process of fertilization, and that's why they are known as post-fertilization events. The events in sexual reproduction which are taking place after the formation of zygote, that is after the fertilization step, they all are known as post-fertilization events. So here it consists of two different events occurring in post-fertilization, that is zygote formation and embryogenesis. So here the first event is zygote formation. So after the process of fertilization, that is after the fusion of gamete, the next step is formation of zygote. When male and female gamete they fuses with each other, it leads to the formation of zygote. So zygote formation is the first event which is considered under the class of post-fertilization events. Now, formation of diploid zygote, it is a universal process which occurs in sexually reproducing organisms. Now friends, as you are knowing that male and female gametes which were been produced earlier, they were haploid in nature. So both when male and female gamete they fuse together, when both haploid gametes are fusing together, they lead to the formation of a diploid zygote, and it is a basic process, a universal process which is occurring in all sexually reproducing organisms. So first point is formation of diploid zygote. So diploid zygote is formed by fusion of haploid male gamete and haploid female gamete. So this leads to the formation of a diploid zygote. Now, as we have gone earlier, that there were basically two different types of fertilization which were taking place. One was external fertilization, which was taking place outside the body of the organism, and the another one was internal fertilization, which was taking place inside the body of the organism. So in case of organism in which external fertilization was taking place, means fertilization was taking place outside the body, the zygote is also formed in the external medium. Usually it is water. Whereas in those organisms which shows internal fertilization, in those kind of organisms, zygote is formed inside the body of the organism. So depending upon what kind of fertilization is taking place in the body of the organism, formation of the zygote also follows the same path, that is either taking place inside the body of the organism or it takes place outside the body of the organism. Now further development of the zygote after its formation, it depends upon life cycle of the organism and the environment or habitat in which it is exposed. So there are two different parameters, or we can say there are two different factors which affects the development of the zygote. One is life cycle of the organism as well as the environment where it is present, or the habitat where it is present. In organism belonging to the class of fungi and algae, zygote it develops a thick wall and it forms spore known as zygospore, and this zygospore is resistant to desiccation as well as damage. So in fungi as well as algae, zygospores are formed, and these zygospores are thick walled spores. They are resistant to any kind of damage. Now these kind of spores they undergo a period of rest before the process of germination. Now In organism with haplontic life cycle, zygote divides by the process of meiosis, because zygote is diploid in nature, and the life cycle which the organism it is following, it is an haplontic life cycle, means which would be a haploid life cycle. So zygote undergoes division by the process of meiosis, and it leads to the formation of haploid spores known as zygospores. So algae and fungi, they are the organism with haplontic life cycle where zygote divides by the process of meiosis, leading to the formation of haploid spores known as zygospores, and these haploid spores will grow into haploid individuals. Whereas if the organism or animal it has diplontic life cycle, in that case zygote does not take any kind of rest, which we have seen in earlier case, that is organism with haplontic life cycle, zygote undergoes a period of rest. The same stage or same phase of rest, that is the resting phase, it is not seen in case of animals with diplontic life cycle. The zygote it starts dividing by mitosis, and it will form a diploid embryo, and from this diploid embryo a diploid organism is formed. So zygote is a vital link which will ensure the continuity of species. So this zygote it leads to the formation of a new organism. So it is a link to the continuity of further species, and it leads to the development of another generation. So every sexually reproducing organisms, including human beings, they begin their life with a single cell which is known as zygote. So here, friends, the most important thing to understand in this case is the development of zygote depends upon the life cycle of the organism. Means if the life cycle of the organism is haplontic, then zygote will undergo meiosis and it will form haploid spores. Whereas if the organism is following a diploid life cycle, in that case, zygote is also diploid, and further development of the zygote will be also as per diploid life cycle. As a result, zygote will only undergo mitotic division, and it leads to the development of a diploid organism. So the important thing which we need to understand in this particular point is, if haplontic life cycle is there, then a diploid zygote has to form haploid spores by meiosis. Whereas if it is a diplontic life cycle, then diploid zygote it has to undergo mitosis to form a diploid organism. Now moving towards the another event in the case of post-fertilization event, that is embryogenesis. Embryogenesis is the process of development of embryo from zygote is known as embryogenesis. Genesis means formation. Formation of embryo, it is also known as embryogenesis. Now embryo is formed from zygote. Now during embryogenesis, zygote will undergo cell division, that is mitosis, and cell differentiation. So in embryogenesis, there will be two stages. The first stage will be cell division and the second stage will be cell differentiation. Cell division, it always increases the number of cells in the developing embryo. So friends, in cell division, there will be only increase in the number of cells which leads to the development of the embryo. And the second phase is cell differentiation, where there will be a group of cells which undergo certain modifications to form specialized tissues and organs, which leads to the formation of an entire organism. So there will be, in embryogenesis, there will be two steps. One is cell division and the another one is cell differentiation. In cell division, mitosis will take place and large number of cells are formed. Whereas in case of cell division, group of cells, they undergo specialization, they undergo some modification, they will become some special tissues and organs to perform some specific function, and this process is known as cell differentiation. Now, the animals are categorized into oviparous, viviparous, and ovoviviparous. These are the three different classes in which animals are characterized depending upon the development of zygote taking place outside the body of the female parent or inside the body of the female parent. So, how the development of zygote takes place, or where the development of the zygote takes place, either inside the body or outside the body, depending upon that, animals are classified into three different types: oviparous, viviparous, and ovoviviparous. That is, either they lay fertilized or unfertilized eggs, or they give birth directly to the young ones. Now, in ovoviviparous animals like reptiles and birds, fertilized eggs, which are covered by hard calcareous shell, they are laid in safe places in the environment. So, fertilized eggs are laid, and after a period of incubation, the young ones will hatch out of the eggs. On the other hand, in case of viviparous animals, so in viviparous animals, that is a majority of mammals, including human beings, zygote will develop into a young ones inside the body of female organism. So zygote will be developing inside the female body. So in humans also the same process takes place. That is, zygote, it develops inside the body of female, and after a certain stage of growth, young ones are delivered out of the female body. That is, in case of humans, after 9 months of growth, the young ones are delivered out of the body of the female organism. Now here, in case of viviparous animals, there will be a proper embryonic care and protection which will be taking place, because the zygote is developing inside the female body. A proper embryonic care and protection is given to the young one. As a result, the chances of the survival of the young one, it is far better as compared to in case of oviparous animals, because in case of oviparous animals, they are more uh, they are more prevalent to the damage by the predators. So predators, they can try to attack these kind of young ones in case of oviparous animals, of reptiles and birds. But in case of viviparous animals, it is given a proper care inside the body of female, and there are more chances of the survival of the young ones in case of viviparous organisms. Now, in case of ovoviviparous animals, as the name suggests, that partially they are oviparous and partially they are viviparous. So here what is happening, that the females will retain the eggs inside the body rather than hatching them out as it occurs in case of oviparous animals. So in oviparous animals, the eggs are basically, it is laid outside, whereas in case of ovoviviparous animals, the eggs are retained inside the body. So eggs will be present inside the body of the female, and the embryo development takes place inside the body of the female without any kind of any extra nourishment to the developing embryo. So to the embryo there is no extra nourishment which has been given. So development of the embryo takes place inside the egg which is present inside the female's body, because such kind of organisms, they do not have placenta, and that's why they are not able to provide nutrition to the young ones. So in case of female animals of ovoviviparous class, they give birth to the young ones. That is in case of sharks and rattlesnakes. So partially they are oviparous and partially they are viviparous, because they will form the egg, but they will retain the egg inside their body, and development of the embryo will also take place inside their body without any kind of extra nourishment given to them. Now, in case of plants, zygote is formed inside the ovule. So, zygote formation takes place inside the ovule, and after the fertilization, the sepal, petals, and the stamens of the flower, they fall off. Now, hibiscus is an example of a plant in which the sepals, they remain attached even after the process of the fertilization. The pistil, however, remains attached to the plant. The pistil, that is the female reproductive organ, will be attached to the plant, and the zygote develops into an embryo. ovules will develop into seed, and ovary will develop into fruit. So in flowering plants, a zygote develops into embryo, ovule develops into seed, and ovary develops into fruit. Now after the formation of fruit, there will be a thick walled formation which is protective in function, known as pericarp. This is an important question with respect to examination. What do you mean by pericarp? Pericarp is a thick-walled protective cover which is formed outside the fruit. It is known as pericarp. Now, after the dispersal, the seeds will germinate under favorable conditions, and they will also produce a new plant. So here there is a diagram which shows various kind of fruits along with their seeds, where you can see that the letter P indicates pericarp and S indicates the seed. So there are various different examples of the fruits along with the seeds, where you can see presence of pericarp, which is light yellow in color, and there is presence of seed which has been given as a greenish color. The greenish color is the seed, and the outer wall which is protective in function, that is the pericarp. So with this particular video we have completed chapter number 1. From the next class onwards we will start with chapter number 2, that is reproduction, sexual reproduction in flowering plants. Thank you.